deep sea. There's likely to be a hell of a lot more species than that out there. That's just what we've already got. A third of these are deep sea species, half of these are deep sea species, and all of these are deep sea species. I know that it's quite easy to get jealous, and it's quite easy to recognise this family, but I wouldn't expect a non-expert to be able to get further than jealous, because it's too difficult and the keys don't exist. The Lepidolinae, quite a distinctive amphipod by these spines, dorsal spines all the way along. There's 38 species in 4 genera, and a lot of those are deep sea. And these are 2 known recent revisions, but this one has information about the generic distinctions, and this one is a South Atlantic key, so it's not very useful. You would end up probably having to go back to original descriptions in order to get further. But you could get to the generic level here, because this is quite a distinctive group. Another very pretty distinctive group, and they're characterised by this dorsal carination. There's only about 25 deep water species in Echimeria, which is one of the genera, and then there's just this one monotypic, very recognisable species. And this is very well covered, the identification of these animals, by Coleman 2007, and I thought we have this available, and it's a very good reference, and this is the one that also has the morphology of the amphipods covered at the beginning. Another closely related group, the same reference, and they're quite difficult to identify because it works on mouth parts, and that always frightens most people off, it even scares me. So you have to really decide whether you're going to be likely to need to identify to a species level of these. There's not going to be that many of them in your samples anyway. Eusauridae are very, very common in the deep sea. This is a definite deep sea group. There's 11 genera, 115 species known, 8 of these genera and 80 species are deep sea. And they are well represented in the CCZ. The specimens that I've received consist mostly of this group. And they are mostly this genus here, Rhacotropus. They are most likely to be new species. And certainly in the sample set that I had at least 2 species, 2 new species of Rhacotropus. So this is going to be a problem genus that we will need to work on getting some operational taxonomic unit sheets prepared. And that's very doable. There are lots of characters on these animals, and they're quite big, and they're quite easy to recognise the different characters. So you could certainly produce a key to the Rhacotropus species of the CCZ without too much work. These are the major characters that you use to recognise Eusauridae from other amphipods. Calliopeidae, there's a lot of deep sea species, but it is mostly a shallow water group. So there's about 27 deep sea species known, and there's actually 102 species overall, so it's not a natural deep sea family. Similarly here, there's a deep sea family that are within this group that are all deep sea, and they are keyed in this lovely Sousa, Filigu and Sorrego in 2014 keyed out all the deep sea species in the most important group in this group. And they're characterised by this Uropod 3 structure. Leucothoidae, you could come across them, they're mostly associated with sponges and corals, and they're quite characteristic, and there is a very good means of identifying these groups. They've even got, I think, an INT key online that's available for them, so if you do have any, there is a possibility to identify them, again, likely to be new. These 2 groups are very tricky. There's a lot of species, 344 species, 150 species in these 2 genera, but most of them are shallow water. So any that you do come across are likely to be new, but can be identified from the few deep sea species that we know about. So whether you don't need to be overwhelmed with the diversity of these animals in the first place, because the actual number of deep sea species that you need to compare it against is very few. Pardaliscidae are a characteristic deep sea group. We have got a lot of these from what I've seen of the samples. These are found in very easily in both these and the eusyrids, found very commonly in the epidendric sledge material, because they tend to live in the epidendric layer, swimming around carnivores. There's 22 genera and 76 species. Most of these genera will contain a deep sea representative, but most of them, there's only one species in each genus, so that's what I mean by monotypic. But Pardalisca, Alisi and Hydroides, there are lots of species in those genera, and a lot of them are new or undescribed, and there isn't a key. Similar situation with the Cerotidae. These are going to be very common in the deep sea. This is a family which is easily recognizable by the characters which I'm pointing out here, and which is provided in the main part of my talk, my summary. And these are the texts that you would use. But it is a very difficult group. It's very diverse. There's a lot of species found in the deep sea, and a lot of them will be new. It would be very good to get to the genus. Foxocephalids, as Charlotte mentioned, another difficult, diverse deep sea group. There's been 7 subfamilies described, and deep sea representatives are in most of those. You need to use the key here in Barnard and Karaman, which is the global key, to get through those. And certain groups are more important in the Atlantic, so these 2 genera are very important in the North Atlantic. And I've seen Leptofoxus and Leptofoxoides are important in the Pacific. They are quite well represented in the material that I've received. This is a true deep sea species, quite recognisable by the large deep coxy. There's 100 species and 26 genera, and they're quite difficult to identify because they do use mouth parts, and they're very quite brittle animals, and as soon as you start taking off these coxy to get to the mouth parts, they tend to fall apart, so it's not easy to work with these ones. Similar situation with here. These are the stenopholids. There's a lot of species in a lot of genera. They are very, very tiny, and it does rely on mouth part use in order to identify the genera. So not easy, but they shouldn't be too common in the deep sea. There'll be some. This is a common deep sea group, Cynopiidae. There's many deep sea species, and there is a key to all genera and a key to all species. 2013, this is a recent one here. It's very useful, and it has a very good description of the morphology of this group and how to identify it. I'm starting to move into the Lycaenassoidea, and that's where we have a real problem. This is one family within the Lycaenassoidea that we can understand. There's deep sea species in the genus Aristeus, 15 of them are deep sea, and we have a short coxa 1, shorter than coxa 2. And this is quite a recognisable group of Lycaenassoids. But the story doesn't end there. The Lycaenassoidea are a very large group. There's currently 22 families containing 177 genera and more than 1,000 species. And these are very, very common in the deep sea. And this is where our big problem lies with the Amphipoda, because while you would call them Amphipoda, we get to Lycaenassoidea and stop there, and that's where it's very difficult. And that's why Charlotte's been doing the molecular work on this particular group, because we have some very unwieldy, very difficult taxonomy here. These are the 2 major families, the Lycaenassoidae and the Euristidae. As you can see, this contains 500 species and split subfamilies, and there's no good keys really for these. So we do have big problems with the Lycaenassoidea, but both myself and Charlotte are experts on this particular group because that's where the problems lie. So how do we proceed with identifying these sorts of difficult taxa? They are very diverse at the family level, and we have some difficulties when we receive specimens with a lack of diagnostic characters. For example, if you see the pictures that I've given you there, a lot of the characters, all the characters are needed in order to get us to that place. So if they lose some legs, or they lose a head, then it makes it very difficult for us to identify them. So we can get, sometimes with an expert eye, we can get to family. But without an expert eye who has the experience of the Amphipoda, it'd be really, really hard even to get to that level. Some of these identifications do require dissections of mouth parts, but again, this depends on the family. You should be able, that's to get beyond family, will require dissection of mouth parts. ways, and if we can focus on getting diagnostic character state sheets prepared for each of these families, I think we have a way of proceeding. So each family can have the diagnostic characters for that family available. We can then produce sheets which could be shared between contractors and samples shared, and we build a picture of these operational taxonomic units. And I think this is the way we have to proceed, particularly with these small taxa. I'd like to thank KIOS for hosting the workshop and the ISA for supporting our participation.
Thank you very much, Tani. A question, please? Yeah, Lenak.
Do you know the reproduction strategy of antipodes?
It's the same as the Tanaids. They brood in, they lay their eggs into a brood pouch and they release them. But unlike the Tanaids, it will vary by family and by suborder. So we have pelagic species, we have epibenthic species, we have the scavenger species which are travelling up into the water column and then they're releasing their larvae away from a bait. So it's much more variable how we will understand the dispersal and the species ranges. It varies within the Amphipoda.
And so do you know studies that have been looking at dispersal ranges for different kinds of species or functional groups?
Probably more the sort of work that Charlotte's been doing on the molecular work with the scavenger species. We have looked at that sort of thing. So you're finding that they're dispersed differently depending on depth, but these are the well-dispersed species, the well-dispersed groups. For the informal groups, some of those families, I can tell you, I would say that family is informal, and that family will have a dispersal like the Tanaids, very unlikely to go very far, and they don't swim up into the water column. But that would depend on, the ecology depends on our knowledge of the group. Yeah. It is available.
But for these families that you suspect have very low capacity for dispersion, do you know some studies that have checked that? Or,
So.
Thank you very much for your overview of Amphipod family. And you don't mention about complete isopods. Did you find them?
Caprilis.
Yes.
I haven't seen any within the very small sample set that I've received, but although they weren't mentioned specifically as a major family, they are within the Centichordata now, so they're within a different suborder. So they seem to be they've become more hidden within the Amphipoda. So they were in that talk, but hidden now. They're part of a different suborder. But we don't find very many, but they will be associated more with epifaunal. So they tend to attach.
Any other question?
Yes. I like questions.
Someone's going to take my microphone off.
Okay.
I'm starting to get, and I'm a bit late again, sorry, but I'm starting to get an understanding of how complex this is. And from a contractor's perspective, I'm starting to see that from a single box core, we may need to be sending parts of that sample to different places, to different experts, for different types of taxonomic identification.
Yes.
And I'm wondering what, in a general sense, in your next talk might talk to this, so feel free to park it, what the logistics of that might be, and also what we should factor in terms of timeframes for sending samples all over the place and then getting a consolidated understanding.
I think that's the major part of our discussion going forward. And I think you're right, this is something that I cover in the next talk, but that's not till Saturday, so we can discuss it a little bit here now. Is that yes, I envisage that we will work along a method of contractors identifying material to a level, to a certain level, and then passing them on to the experts for the next level. And I think that's the only way we can really work. We can train like I'm training now, or we'll be training over the next few days, to identify to that certain level. But the final step really, because of the complexities of, as you say, the diversity of each of these groups, the final level really does does have to be done at the expert level. How we manage that, I think, is how we have to plan now. We have to plan databases, we have to plan collections, references, reference materials, and how we're going to share those. It will change the timeframes, but it could speed things up rather than ending up with these animals just parked as amphipoda, meaning nothing going nowhere. So I think it's the way we have to work.
That's a good question. Dave?
So that has some cost implications on how we organise ourselves. Is the idea then to have a list of, if you like, an expert working group for different taxa, and then is there something that we are going to have to be discussing before we get to Saturday, but maybe the major discussion on this would be on Saturday, would be then how that is going to be funded. If we're going to have a group that aren't necessarily related to contractors, but which obviously would be servicing, I suppose the contractors then would pay for those services in some way. But is that the best way of organising it, I suppose is the question, or is there a need for some collective way of funding this type of identification or final identification?
I don't think I'm the best person to actually answer that, but I think we would need, obviously you're right, it does need some funding, it does need a funding model. It could be that the contractors provide a pool of funding to one central pool, and we have a two-stage where the contractors will fund their own cruises, their own identification to that level, and they provide funds to a central pool where we all share the data as a taxonomic clearinghouse, as it were. That may be one of the mechanisms that would work.
Thank you very much, Tami. So we pass to our next presentation by Stephanie Kaiser. Another important group, Isopera.
Following on from previous presentations, this may be a little bit repetitive, so I apologize for this. In this talk I'm going to focus on this topic, that is practical information on sample methods for processing as well as methods for species discrimination. Genetic analysis will be covered by Annika Janssen this morning, so tomorrow. So I'm going to focus on morphological investigations, and in the end I will present some preliminary results of the biodiversity distribution of season-dead isopods. So isopods occur both on land and in the sea. About 10,000 species have been described, about half of them in the marine environment. They are particularly diverse in the deep sea. About 3,000 species have been described from below 200 meters. They show a variety of body forms and feeding types, so from suspended feeders, detritus feeders, predators. Most deep sea isopods are detritus feeders, and some have been shown to be feeding forms. Apart from parasites, all isopods are affiliated with predators, like amphipods and canalids, and this may limit their distribution to active feeders. Nevertheless, there are some differences in mobility between types and groups. For example, some families are really good swimmers, such as the Minomycidae. They have highly modified posterior swimming legs, while others have walking legs or follow an informal boring lifestyle. So depending on the phylogenetic system you follow, there are about 10 different suborders, and these the ones pictured here, they have all found in the abyss. The isolated isopods, they are the most dominant isopod suborder. About 90% of specimens belong to the isolotans, while the other ones are much less frequent. For example, the spermatozoids and the wild ferns, they have the greatest diversity on southern hemisphere shells, whereas the chromathoids particularly diverse in the tropical and the shallows. But at least representatives of 3 of them are still chromathoids and spermatozoids are found in CCZ collections. So in CCZ, some families are more diverse than others. The most abundant ones, they are typical deep sea families with a cosmopolitan distribution. They are isolated greatest species of diversity in the deep sea. Normally, the family Monoxide is the most abundant and diverse group in deep sea collections. For example, it provides much picture samples from the North Pacific and Southern Ocean rivers. The Desmosomatida, they are the most dominant, and it's not clear if this is a real trend or just something wise. So where did the samples we work with come from? So at Lenkenberg, we mostly work on samples from the bench classes area and the germ classes area, and some of the registry forward expeditions in 2010 and 2014, and the identification of isolate species is based both on morphological and genetic methods. And these data are then used to assess diversity and distribution in framework of large spatial scales. So this has been mentioned before. So for isomer collection, we also use mostly the equivalent sledge and the box scorer to a lesser extent. The advantage of the sledge is that it samples more specimens per sample unit and compared to the box scorer, but in contrast to the latter, just provides qualitative samples. So the sledge is equipped with 2 nets on top of each other. That's an upper supranet and a lower epinet. And the initial idea was to sample different faunas, but apparently this is not the case, so usually sample separately and in the end merge data from both nets. The doors here are closed, there's a level mechanism, so the doors here are closed, and they open as soon as the sledge is the seafloor. And for genetic analysis, it's important to keep the samples cold at all times. So for the ground and tropical waters, the proteins are surrounded by a cooling box, which saves the deep water from the deep, and so the samples stay cold until they are retrieved for further processing. So we usually preserve samples for both morphological and genetic analysis. So after retrieving the cod ends, the samples are iterated with cold seawater and then sift through a 500 and 300 micron mesh. The samples are then fixed in pre-cooled ethanol. The fixation in gas is also possible and possibly much cheaper